Alright guys, so Kember is back. I'm back from my hiatus. As promised, here is the science behind my honors thesis. For any of you that didn't see my previous upload, which was just a recording of my thesis defense essentially, for those of you who don't know, thesis defense, that is for the past, now it's like 4 years of my life, I've been working on this specific project with this principal investigator at the university I go to. We've been working on this project, and at the end of my time, since I've graduated now, I basically summarized all of what I've done and presented it to a committee of 3 professors where they look at all of the things I've done. They deem me either a good or not researcher. Did I do the due diligence I needed to come up with the results that I did from my work? And I uploaded that, but I wasn't really super satisfied with it because my whole goal on this channel is to try and reach an audience of people who don't necessarily interact with biological stuff too often. And I think it's a very steep entry curve even for somebody who does have that biological background to watch my thesis defense. So here I'm going to take it a lot more slow. I don't have a 30-minute timer of making this as short as possible, so I can ramble for as long as I want to and try and really decode what my project is for a layman, so anybody can really understand, just because I really do enjoy my research, and I hope that everybody has the joy of understanding the thing that I've been working for the past 4 years. And if you haven't seen my past video, my research topic is mainly focused in structural biology, and it's a structural analysis of OSC1 binding to nicotinic acetylcholine receptors using cryogenic electron microscopy. So it's kind of a handful, probably don't recognize a lot of those words, but I'll work through it in due time. All you really need to know for right now is I've synthesized this specific toxin, and I'm visualizing how it binds to this receptor using electron microscopy, giving atomic resolution, as seeing how this toxin causes paralysis. So that is the overall theme of my project, and I've cut it up into a couple different sections, so if you'd want to skip over, I have that listed. But basically the sections that I got here are initially just going over what is OSC1, what is this toxin, how does paralysis work, how does motion work, then the actual production and purification of this toxin and the receptor, then delving into what is electron microscopy or cryo-EM, and then lastly going through the data analysis, and I also have a little bonus section at the end. Well, let's just dive straight into it with what is OSC1 and how does paralysis happen. So starting off, OSC1 is made by this cute little scorpion here. Scientific name is Orthochirus scribiculosus, and the common name for the scorpion is a Central Asian scorpion. And basically I'm making the toxin that this scorpion naturally produces because it has a good ability of being able to bind acetylcholine receptors and cause flaccid paralysis. That's what this toxin does. And just looking at molecular structure here, you can see in green, this is the structure of the toxin. This is just showing the backbone with kind of a ribbon structure, so you're not seeing any of the functional groups of the amino acids. So if you're looking at the amino acid structure, you have this nitrogen as well as this carboxyl, and then at the middle there you're going to have this hydrogen and then an R group that's facing off. So in this case we see only the backbone, no R group on this. And just kind of giving it the general structure, we can see there's this spirally structure that's alpha helice. You have 3 beta sheets. And this peptide is made up of many different amino acids. So in our case, OSC1 has 38 amino acids it's made from, of the 20 canonical amino acids, which I'm showing on the screen now. As you can see, some of these amino acids have very similar, chemical attributes. Some of them are hydrophobic, some of them are polar but uncharged, some of them actually do have a charge, and some of them are just kind of weird, have special use cases, which in this case, if we're looking at cysteine, this amino acid is actually very special in the case that these sulfurs can if there's two cysteines in a peptide, they can interact with each other and create a disulfide bond where these two cysteines kind of lock themselves to be next to each other and limit the amount of conformations that protein can get into, I guess, for lack of better terms. Just looking at the primary structure of this protein here, what we can see here, the 38 amino acids laid out in the specific order, that if you have these amino acids laid out in this specific order, you're going to get OSC1, a toxin that causes flaccid paralysis. And you can see there's 6 different cysteines in here. And you can see the way they're supposed to bond to each other with those bars there. And we can look at a 3D model of what OSC1 actually looks like here. Just spinning around, this is showing the backbone and ribbon form, and then also showing the functional or the R groups, which you saw earlier. Here, all the R groups are shown. You can see, while it's spinning around, you can see in the middle there, you can kind of see something yellow bonding to each other. Those are the disulfide bonds between the cysteines there. You can see anything that's blue, it's going to be nitrogen, red is oxygen, tan is carbon, yellow is sulfur, I don't know if I said that already, and then the white is going to be hydrogen. So I was basically showing all of those amino acids, how they pack up next to each other. If we're looking at a protein, you need to think about its structure, because its structure is heavily related with its function. So in the previous slide, I showed you the primary structure, which is just the amino acids laid out with which are needed to make OSC1. But then you also have secondary structures, which are basically just energetically favorable ways for amino acids to interact. And they're so prominent that they're called motifs, or basically repeating forms of ways these amino acids interact to stabilize the structure. So you have alpha helices and beta sheets, which chemically look different, but they're stabilized by hydrogen bonding, something which I will go more in depth to once I start talking about results. But you have these hydrogen bonds that stabilize these alpha helical and beta sheet structures, which further give some sort of form to this peptide. Then from that, when you have those structures, you can go into tertiary structures where you have hydrogen bonding in between 2 different amino acids, where they aren't creating that, I guess, alpha helical or beta sheet structure, but it can contribute to the way in which where different amino acids would like to be in orientation with each other. You can also have salt bridges, where if you have charged amino acids of opposite charge next to each other, they might attract, or if they're of opposite charge, they might repel, and so on and so forth. And then lastly, we get quaternary structure, which you can think about this OSC1 is its own peptide, it's its own protein, and it's going to interact with the receptor, which is a protein of its own. So when these 2 proteins come together and interact, they're forming a quaternary structure, and those interactions between the toxin and the receptor are what I'm really interested in today. And so before we really go into how paralysis happens, it's interesting or necessary to first understand how neurons work. So looking here at this GIF, I guess, when you're looking at the left side, what you see is this squiggly stuff on the top. Those are dendrites. Those are going to be receiving signals from other neuronal axons. And this circle is signifying the cell nucleus or the soma of that neuron. And what happens if the cell body gets very excited it'll get depolarized. And when I say depolarized, I just mean that there's going to be an influx of sodium atoms that get onto the inside of the cell and cause the membrane potential to go from from negative to slightly less negative because of that influx of sodium ions. So basically, when the cell body decides that it's time to fire, you're going to have a rush of sodium ions going into the axon. So you have this wave of positive charges flowing along the axon. And once this wave of charges reaches those branches at the very end, the axon terminals, what happens is these charged sodium particles or ions will cause neurotransmitter to be released from those axon terminals onto the dendrites of some other neuron. So on the very right, these axon terminals are going to they're going to be interacting with other dendrites of other neurons. And when we're looking at specifically when you want motion to happen, the signal originally happens somewhere up in your brain, goes out your spinal cord, and lands, or this axon terminal is gonna hook onto the dendrite of some other neuron in the periphery. And if it depolarizes enough, there's enough sodium going into that second neuron, the cell body might be like, hey, there's a lot of sodium in here, I better fire as well. And that's gonna cause more sodium to flow down into the secondary neuron, and then you get this wave of sodium which is going to show up to the neuromuscular junction, which, looking at a blown up version of this, you can see the neuromuscular junction. You can see that the box there is showing the axon terminal, I guess, in close proximity to the muscle fiber. And what happens, as I said earlier, when you have that influx of sodium at those axon terminals, this isn't exactly what happens. There's more nuance to it, and I'll cover in a future video how neurons fire. But this influx of sodium basically causes neurotransmitter to be released across the synaptic cleft. In this case, neurotransmitter can be something like acetylcholine, a small molecule which can flow across and bind to acetylcholine receptors. What these receptors do, they're found also on this neuron's dendrites, which are currently interacting with the muscle. These acetylcholine receptors, when they are bound to two acetylcholines, I should probably just do this. If they're bound to two acetylcholines, the channel is going to open and it's going to allow sodium to flow into your muscles. So what this does, when you have sodium inside your muscles, in order to not overly complicate it, and it essentially causes a release of calcium from sarcoplasmic reticulum, which then this calcium can then interact with a motor protein, myosin 2, which this motor protein uses ATP and also requires calcium in order to move. And just showing a GIF here of what's happening. Once you have this calcium that's released because of the sodium that God flooded in because of the opening of these receptors, this causes the myosin to walk along these actin fibrils, which actin is a part of your cytoskeleton, so it's used in a lot of other parts of, and basically every cell type is used for a lot of different things, but in this case is used in sarcomeres, so that you have this myosin that's walking along the actin filaments and able to produce a force. And so in order to see how it actually contracts your muscles, we have to look at a sarcomere, which is basically just a massive collection of myosin heads and these actin filaments. And what you can see is when you have a lot of activation, a lot of acetylcholine, a lot of sodium coming in, a lot of calcium released, that lets these myosin heads do work, and they can move, and they can contract the sarcomere so that it becomes shorter, and that eventually leads to you being able to contract your muscle or do whatever it is you want to do. So the nuance here is even if your body wants you to do some sort of motion, you can have your neurons send a signal, there's a flood of sodium that's going down into your spinal cord, comes out, it lands onto some neuron in your periphery. There's a lot of sodium being tossed out, so it's like, okay, I should probably activate too. This neuron then, it has like a lot of sodium which is flowing through it, and let's say I want to move my arm. There's a lot of sodium, or this sodium causes acetylcholine to be released onto my muscle fibers. It binds the receptors in my muscles, and sodium flows into my muscles. That causes this whole thing. But the toxin binds these receptors and prevents those channels from opening. So there is no sodium going into the cell, no calcium is released, and myosin doesn't do anything. So even if your body sends a signal that I want to move my arm, because this toxin is preventing the channel from opening, you don't get any motion even though your body wants to have motion there. And so looking more at the structure of this receptor itself, as I mentioned earlier, there are two binding sites on this nicotinic acetylcholine receptor. If you have an agonist binding, an agonist, very loosely speaking, is just something that promotes the natural tendency of that protein. So in this case, an agonist would allow for the receptor to open and let sodium pass through and let that relay of that signal continue being passed on. But there's also other types of agonists, like, for example, nicotine, which is how nicotinic acetylcholine receptors got their name. We noticed that nicotine is able to bind and keep this channel open. There's also antagonist, which is what OSC1 is. It binds to the same sites that, or it binds to a similar spot to where acetylcholine does, but it prevents that channel from opening. So look, this is just like a cartoon, I guess, image that I was showing you guys of this receptor and showing how sodium goes from the outside of the cell into the cell. Now let's look at an actual, I guess, molecular look at it. What you can see, I don't know if I said this yet, there's 5 different subunits, so, and 2 of them are redundant, so there's 2 pinks and there's 3 other colors. And then you can see there's also a bunch of like these red dots which correspond to the oxygens and sugars and other fatty acids which associate with this receptor naturally. So here we're looking at very small scale. This is 16 nanometers tall by 8 nanometers wide at the top, I guess, when you're looking down the channel. But when we are looking down the channel, let's just look at, here we have a better view of what's going on. Down the middle there is where you'd see sodium ions going through if you had 2 acetylcholine receptors, or 2 acetylcholine molecules binding to this receptor, cause that channel to open, sodium flows through. Something else we can notice while we're here is that there's 5 different subunits here. There's 2 pink ones, which are the alpha subunit. One of them is green, which is delta. The weird looking purple is gamma, and then we have beta. And so acetylcholine binds at the interface in between the alpha delta and the alpha gamma site. And yeah, the toxin will also bind at these interfaces and prevent that channel from opening, essentially. And so just going through one last time to make sure you fully understand what's going on. When your body sends out your brain sends out a signal, it wants to start moving. Something in your motor cortex is like, hey, let's initiate movement. It sends out a signal, which is really just a wave of sodium ions. It goes to the axon terminals of that neuron. It fires a neurotransmitter at the dendrites of some other neuron. It continues the signal, goes down that second neuron's axon, and we reach the axon terminals where it would stimulate the muscle. But if you have this toxin, even no matter how much acetylcholine you have released at this synaptic cleft, it doesn't matter because this toxin is going to prevent, and it binds to more tightly this receptor than acetylcholine does. And it just prevents this channel from opening. So if your body wants to move, for example, if your body wants to move your diaphragm to keep you breathing, even if it's sending those signals, if this toxin is in your body, it's going to prevent that contraction of your muscles in your diaphragm, and you're eventually going to die because your muscles can no longer do their normal function, even the ones that you normally don't have to think about using, like your diaphragm or your heart. So obviously pretty scary. You don't really like to be paralyzed, but there are some specific moments in your life where you would really hope to be paralyzed at some point, and that is if you're undergoing surgery. And this is really the whole shtick of why this research is important, is that there are certain points in life where you do want to be paralyzed, and that's a very valuable thing to have. But this research kind of is more going into studying the state of how you can close these acetylcholine receptors, which acetylcholine receptors are used to drive all of the motion that happens, as well as other bodily functions in your brain. But it's important to look at these peripheral nicotinic acetylcholine receptors to improve our knowledge about paralysis. Because if you're ever, for example, undergoing a procedure where you need a breathing tube, it's very common practice to numb your throat with a neuromuscular blocking drug, which, as the name suggests, it is a drug that blocks action at the neuromuscular junction. So it acts in ways very similar to this toxin, except most of these are small molecules. But essentially, these neuromuscular blocking drugs block your throat from being able to move so that you aren't choking on your breathing tube while you're undergoing surgical procedures. And there's many other reasons why you'd want to use a neuromuscular blocking drug. I'm not a medical expert, so I won't really delve too deep into that, but there are times where doctors want you to be paralyzed for some reason, and there's many different drugs they can choose from. As I mentioned, they're neuromuscular blocking drugs. Looking at this diagram here, we can see that underlined acetylcholine there in red, and also circled this quaternary amine, which is basically just that region that I circled is a nitrogen bound to 4 other things, and because of the way nitrogen is, it will have a formal charge of +1, so it's going to be positive. And as it turns out, this quaternary amine is very important and key to the binding of acetylcholine to a nicotinic acetylcholine receptor. So a bunch of these neuromuscular blocking drugs, if you just look at all the circles that I've made, they also have a bunch of quaternary amines, and they're using those to mimic the binding of acetylcholine to its receptor. And the thing about neuromuscular blocking drugs is we know how they bind, which is that they bind similarly to acetylcholine, but we don't really, we're kind of using the same trick multiple times. So the whole point of this research is, if you're looking at some of these neuromuscular blocking drugs, or even acetylcholine on its own, an amino acid on its own is roughly the same size as acetylcholine. So this toxin that's binding is almost 38 times the size of acetylcholine. So it can bind to many different parts of the receptor at one time and basically scan the surface to find other ways in which we can bind to the acetylcholine receptor, potentially find new sites for binding, and create other small molecule drugs for these other sites to create therapeutics with other types of, usefulness cases. So that is part of the reason why this research is important. But before we fully dive into what's going on, I'm also going to talk about AlphaFold, which you may have heard in the news recently. It won Nobel Prize, I think a year ago or something. Very cool. But using AlphaFold, we were just kind of interested in the paper with which we came to find this OSC1. It shows that it already binds to nicotinic acetylcholine receptors, human and electric ray. All the nuance of that I'll explain later. But it's known that this toxin can bind, but we're just interested to see what does AlphaFold think will happen when this toxin binds the receptor. And so what I'm showing you here is an image of the receptor here, as well as OSC1 on the top, which is circled. You're seeing this basically side view and then top view. On the top is OSC1, and on the bottom is a different type of toxin, alpha bungarotoxin. They both bind at the same binding site, which, as I mentioned, is the interface of the alpha delta and alpha gamma sites. And something else that's important to note is that in these predicted structures, OSC1 is less resolved, and you can tell that because it's a yellow color compared to the best resolved structures are going to be a deep blue, then yellow, and then orange. So OSC1 isn't super well resolved. Its predicted local distance different test value is not as good as alpha bungarotoxin, meaning there's less certainty about it binding to those regions. But it's still valuable to still just double check and be like, yeah, okay, like AlphaFold thinks that it binds, which is cool. And just a little nuance, I guess, as to how AlphaFold works. It doesn't take into consideration the energetics of any of the amino acids at any point in the calculation, which is kind of interesting and a novel way that AlphaFold works, is that you basically input the letter sequence of the OSC1, which is 38 amino acids, and then there's 5 different subunits for the receptor, so you put in 500 letter sequences of the amino acids in the receptor, and it thinks, you know, of the many 100,000 structures that are already solved, when we look at these 2 amino acids that are close together, when we look at all these other structures that are solved, what conformation are these in usually? So it's not actually, it's just looking at past structures that we already know how these amino acids are arranged, and just thinking, now that we have this new set of amino acids, what would we think it most looks like based on what we know other amino acids folded to look like? I don't know if that makes any sense, but that's essentially how AlphaFold works, and it leads to, I guess, its use case not being as, it's a cool program, but as you'll see later on, it didn't really predict what happened in the actual binding all too well. Something else that's interesting to point out that I'll just leave as an interesting tidbit is I looked at 4 different potassium channels, which this toxin is also known to bind. These potassium channels are on the presynaptic side of, like postsynaptic would be like the muscle fibers, and presynaptic would be like still on the secondary neuron and where it's dumping the neurotransmitter from. It can have potassium channels, which can dampen the effects of the incoming sodium so that there isn't as much acetylcholine released. And it kind of produces the same effect as blocking these nicotinic acetylcholine receptors. So I just wanted to see how OSC1 binds these, and for the channel with which it has naturally the highest affinity towards, you can see that instead of binding at the top of where the channel usually opens, like they did for all the other ones, you can see the toxin is actually right in between, like in the very middle, blocking the channel where everything would actually go through, which is an interesting, I guess, foreshadowing of what's to come. But that was kind of a long spiel, but now we can actually get into how I made the toxin itself and how I purified it and all that good jazz. Okay, so now that we have a pretty decent understanding of what OSC1 is and how paralysis happens, we can now delve into how OSC1 was actually made. And so, as you guys know, OSC1 is a peptide. It's made out of 38 amino acids. So if you know that specific arrangement of 38 amino acids, you can actually make the peptide yourself without ever having to come across a scorpion, which, very lucky for me. but the process by which you actually understand the primary structure or the sequence of those amino acids, it can happen through many ways, but it can happen using mass spectrometry, which is something that I touch on a little bit later. But essentially, we already know, or somebody already did the work to figure out what the sequence of amino acids is for OSC1, so all I need to do is just string those together to make OSC1. And basically, I'm gonna try and keep this as little OChem as I can, because I know a lot of people don't really enjoy OChem, although I'm quite a big fan. I thought it was pretty fun. But just looking at some OChem with what's going on here, we're gonna look at this picture here. We can see that this amino acid, and it's already bonded to something called a resin. And basically this resin has this free amine which can bind to the carboxylic acid of some free amino acid. You can see the R group there, just a nondescript amino acid. And then that NH, that amine group from the first amino acid has a FMOC protecting group, which then you can take that off. The protecting group is really just there so that nitrogen doesn't interact with or form bonds with something you don't want it to. You only want it to bond to the carboxylic acid of the next amino acid you're adding. So you have that protecting group on there. When you're ready to take it off, you then add the next amino acid. They bind. You're going to continue that cycle for a while because that next amino acid is going to have that FMOC protecting group, as well as some of the side chains that are used or that amino acids have are reactive, so they have other special protecting groups as well. But once you're done, you can see there at the very bottom you have cleavage, which basically happens using this really strong acid. It cuts that peptide bond from that first amino acid to the resin, and also takes off all of the other protecting groups that protect the side chains of that specific protein. Okay guys, so editor Kember here. I went over this part and I didn't think I did super awesome, so I'm gonna redo it. Essentially, I found an old schematic that I drew up when I initially joined the lab, where I was drawing out the scheme for how I was making this smaller peptide toxin. And I think it's really beneficial to look at it just to really get a better understanding of how this cyclical process works. So I'm just projecting here. Let's just look at the red line first. That's looking at the resin. And so there's this bigger structure that kind of hangs off of the resin, which you can see with those two benzene rings with those, what would you call that? I don't even know at this point. I haven't taken out chem in a long time. But you have this protecting group on there, the FMOC protecting group, and you can see if we go from left to right, this protecting group, the FMOC protecting group, will react with piperidine, which I circled in pink there. And once you get this resin deprotected, what you can do is then you have the free amine on the resin, which is free to interact with the carboxylic acid of whatever amino acid you add first. So in this case, we're adding proline, and I'm showing that with the yellow circle there, that this carboxylic acid is going to react with that amine. And as you can see, when you go down, when you add the coupling reagent, you lose H2O from that peptide bond being formed, and you end up with a proline there that's attached to the resin. And I also circled in green, that's the FMOC protecting group, and from after this point I just label it as the FMOC protecting group. So you can see the resin actually had its own FMOC protecting group, and the FMOC itself interacts with the piperidine. They make a complex which then washes away, and then you can have a deprotected amine so that it can attack the next amino acid that you add. And that's what you see with the piperidine. You have the FMOC on there, and then you add some piperidine, and then you have the free amine that's free to attack the next amino acid, which in this case is another proline, which you can see I circled the top proline and blue in the bottom one as well. So you can see we have two prolines attached to each other. Now, going into the next page, here I circled some other stuff for you. In those pink boxes there, you can see that's showing off the peptide bond, all of the ones that are shown here. And then in red, I'm circling all of the functional groups or R groups that are present, just kind of letting you track how this thing, I guess, gets extended as we're synthesizing it. And then looking at this last page here, the only thing I really want to point out here, I guess, is you can see off of the functional group of one of these amino acids, glutamine, I circled this tridal protecting group, which I think I mentioned earlier. And this basically just protects that amine from being able to react with anything else and just kind of gives us the certainty that when we add the glutamine, it won't react with anything else and you won't have other sorts of reactions you don't want happening at that site. So we cap it off with the tridal group. And then once you have added all the amino acids you want, then you add this really strong acid, as I said earlier, and then you can cleave this peptide off of the resin. So with that red line down at the very bottom, you can see that's where this is actually gonna get, the proline is gonna get cut off from the resin, and then you have your peptide product, which then you can use, purify, do all that good stuff. But just something I want to mention about piperidine, which is just like a fun little tidbit, I guess, is actually a DEA scheduled chemical. And it's a really funny reason, actually. It happens to be a really good weak base, and it reacts with the FMOC group, as you saw in the very first slide. But it's also very heavily watched over by the government because you can use piperidine to make PCP, no other than angel dust, as some of you may know it. I just think that's a funny quirk of working in peptide synthesis. And whenever I had to reorder piperidine, there's always like some scrutiny of, I had to fill out some like forms or something to be like, yeah, this is official business. So I'm sorry to say, but I'm not a PCP dealer. You guys, if that's what you're here for, I cannot supply. That is totally my bad. Sorry for letting you guys down. Moving on, now we're just kind of showing the process of that's just like a cyclical nature of you're just continually adding these amino acids onto the back of each other. The machine you use for this is a peptide synthesizer, where you can kind of see there's like a bunch of different tubes, or not tubes, there's a bunch of different containers with different reagents. There's also big jugs of other chemicals used, and there's also hooked up to gas and all that. It's just more complex than I'm letting it on to be. But you can see all of these tiny little capsules or vials which are filled with different amino acids, and they're going to be added on sequentially. And the resin is kind of acting as a scaffolding where you can elongate this peptide, and once you have elongated as much as you want, you have the protein you need. You can then chop it off that scaffold, and then you have the protein just by itself. And that's what you can see in this picture here that I'm showing. It's basically that scaffold is getting torn apart from the peptide itself using a really strong acid, and in this case it has a really cool, I guess, orange color to it. The resin itself is kind of like it's like a solid, doesn't really, I don't know, it's like a pea yellow type of thing at the bottom. That's the resin there. But yeah, once you make the peptide and it's already off the resin, the only thing after that you need to do, in the case of my peptide OSC1, since it has 6 disulfide bonds, you need to fold the peptide, or in other words, you need to form those disulfide bonds so that it's in its natural structure. And sometimes you can do it in ways where you fold each of those ones systematically. But what we find, and what was found by people who previously synthesized it, is that there's many different ways in which you can have those, because if you're looking at the primary structure again, there's many, like the C8 doesn't have to bind to C28. 8 could also bind to 14. There's many different ways by which you can have these disulfide bonds form, but we want them in a very specific way. But it just so happens that, at least for a majority of peptides, when they have these disulfide bonds, is that if there's many ways in which they could form, usually the one that just kind of happens naturally is the one that nature uses, because it wants something that it is impossible to get stuck with a different disulfide conformation. So what we did, we just took some of that, we took all of that product we got from the synthesis, just put it in this specific reaction, make sure let it stir overnight, and by the morning it was already completely done. But the thing about, like, once you have that, you still don't have a purified product, because there's still many different side reactions that could happen that make a product that isn't satisfactory, that isn't all just a uniform OSC1. So you need to purify that out in some way. And the way you do it is by using high pressure liquid chromatography, or HPLC. Some people say high performance, tomato, you know. But you're using an HPLC, which on this image here, just looking at this yellow circled John, you have this column, which on the inside is filled with these just like hydrocarbon tails, really. And that's, if you're just looking here, you can see that this is what's on the inside of the column. It's just this scaffold attached to this hydrocarbon tail, which is 8 carbons long. So we'd call this a C8 column. Specifically, when I did my purification, I was using a C18 column, which basically just means there's more, use like carbon bound to hydrogen. Since they both have roughly the same electronegativity, there's no partial charges or anything, and it's all kind of neutral. So neutral stuff doesn't like interacting with water, and proteins sometimes don't really as well either. So what you do is you take this protein, you dissolve it in water, and you flow it through the column. And then in the column there, like all these tails are kind of like, you can have a peptide just go in there, and these tails just holding on to it, and the peptide will like just hanging out there, you know, because I don't know, like, why wouldn't you? You know, you're hanging out with a bunch of hydrophobic tails. Like, that sounds like a great time. Might as well grab a beer or something and just hang out for a bit. But when this peptide is just kind of chilling out there, you can then force it to come off the column by adding acetonitrile into the water, which is just some other chemical. You don't really need to know much about it, other than once you add more acetonitrile, this peptide no longer wants to be like in this hydrocarbon, like whatever you call it. It doesn't want to interact with the column anymore. It'd much rather be hanging out with the acetonitrile in the water, and then it falls off the column. And when we're looking at the HPLC itself, you can see there's 2 different bottles on top. One of them is mainly just water, and one is mainly just acetonitrile. So what first happens is you kind of see towards the middle there, there's that tray where you can load a sample. And this sample is going to be loaded onto the column with basically just water. You're going to have water flow from the orange through the column. And since there's basically only water and the peptides would rather interact with the column, all of the samples stay stuck to the column, and you just have water flowing out the back. But then something that's important to note is that at a specific acetonitrile level, all peptides of the same chemical composition will fall out at the same time. So in the case of my toxin, there's a very specific percentage of acetonitrile in the water where all of it is unanimously like, yup, hydrocarbons, this column, not cool, I'd rather hang out with acetonitrile. And it happens at a very specific percentage of acetonitrile every single time. And so what you do is, this image, when this peptide falls off the column, it goes in the green towards that structure over there, the lamp, which is shining a very specific wavelength of light on all of the passing solution. Something to note about amino acids is that they absorb light at very specific frequencies. So you're looking at all this wastewater that's coming off the column, and you're trying to see when you have absorbance in light frequencies that are more, I guess, relevant to peptides. And that's how you can tell when something comes off the column. And in theory, if you are making a simple peptide, most, if not all, of that absorbance that you get throughout that whole sequence of purification should just be your protein product. And basically, when you get to that specific concentration of acetonitrile, instead of it running through the lamp and then like back out into the waste, you can then collect that, so then you have purified OSC1. And that's how the purification happens, very, I guess, simply put. But you can look at, here's a, I guess, graph showing the purification, which it's looking at 4 different wavelengths, so 214, 280, and then 230 at the bottom. Only a couple amino acids absorb at 280, so that's why it's kind of so weak. But what you can notice, this is the very purified product. This is after all of the purification, and just to verify that it is purified. And you can see there's just one singular big peak, tight peak, which is showing that all of what I had in that sample when I put in the HPLC is all one specific product. So to further confirm that what I have is the real deal, we can use a mass spectrometer. I'm just gonna show a GIF here and just talk you through the steps. Essentially what happens is you take this peptide and it gets vaporized and shot out with an electron gun, like in the ionization chamber, which is basically trying to make this protein become charged, because a majority of, maybe not a majority, but like every protein has a specific, like they're majorly bulk neutral, although there are some regions that do have charge on proteins. But what you're trying to do is basically create a specific amount of charge per mass, so that you're trying to create a charged protein, because then you can use an electric field to accelerate that protein so that it goes really fast, right? So what is the point of accelerating the protein in an electric field? Is so that then you can use a magnetic field to then bend the, I guess you're seeing it on the, here in the magnet, the green light looking John. When you get accelerated through the electric field and then go through a magnetic field, the amount that you curve through the magnetic field is a function of how heavy that thing is. So if you're accelerating everything at the same amount, and then is being curved through a magnetic field, through some calculation, you can then not like retrofit, but you can basically come back to how much your sample weighs. And that is another way in which we can verify that we made the right thing here, which, looking at I guess the data we got back from that when we submitted it for mass spec, you can see that the Z is just the mass to charge ratio, so in this case it's 3. So what that means is you can multiply Z by all of the number, like the relative abundance of the weights is shown based on like how high they are, and if you multiply that weight by 3, that'll give you the like total mass of the thing that you were weighing. So in our case, it came out to roughly 4,200 grams per mole, which is what we would expect of our toxin, so we know it's made correctly. Now, going on to, we have the toxin made, right? And now it's going on to the receptor, which it's also a protein. And looking at the UniProt database, I'm just showing the human ChRND gene, which is looking at the delta subunit of the nicotinic acetylcholine receptor. And you can see that it has it's made up of, that one subunit is made up of 517 amino acids. And because of this reason, because of how big this subunit is compared to the 38 in OSC1, it isn't feasible or possible to produce receptors in the automated way that you can with OSC1. And the main reason is that it's just too long, but there's other reasons as well. And I'll kind of go into a simple explanation here of why, you can't use automated synthesis to make these subunits as well. And that's just looking at this simple function here, x to the power of n. So x is the theoretical, I guess, binding coupling efficiency of this amino acid to the next amino acid to the next. So, and then that's to the power of n, which is the amount of amino acids total in that chain there. So we can just assume, just for simplicity's sake, let's say that each coupling is 99% efficient, which it's a good estimate to say that all of coupling is roughly that efficient, although there are specific couplings that are more or less efficient. But let's just say on average, of any way you could possibly combine these amino acids, roughly speaking, 99 out of 100 times, you're gonna get good coupling. So just looking at the first case here, when we have 0.99 to the power of 1, you get 99% total yield of the thing you wanted, which makes sense. 99% of the time it works out, and you only want, I guess, one thing connected to the resin, I guess, and you only have one case where that doesn't happen, right? So then we can go up to 38 amino acids, which is the length of the OSC1 peptide that I made. And you can see that at that point already, only 70% of the stuff that you cleave off the resin is actually going to be what you wanted to make in the first place. So if you go up to something that's even like 100 amino acids long, and you suddenly get dropped down to below 40% of the stuff that you made is what you want. And once you go up to 520 amino acids, then all of a sudden you're at below like 1% of the stuff that you made is actually what you wanted. And basically the easiest way to explain that this is why we don't do it is because it just simply, there's too much other stuff and there isn't anything notable about that one subunit that we could say, like we can't, I don't know how to explain this very well. Basically, the instrumental capabilities of sub 1% of what you made being the right thing is like trying to find a needle in a haystack. You know that it's in there somewhere, but you need to be able to find that one correct spot, and we just aren't at that point where we can make something that long, I guess. And one other way in which you could make these receptors is by having just yeast make them or some other eukaryotic organism. Is the reason for which there would be able to potentially make these receptors is that yeast also use glycosyl transferases or could potentially use human ones, which these are basically just enzymes which attach sugars onto different things. And as you saw earlier when we were just looking at the structure of the receptor, it has a bunch of sugars on there, which that's another reason why we couldn't do the automated peptide synthesis route, is that it's kind of hard to automatically synthesize it as well as being able to precision point, like put sugars at very specific points that we'd want along there. But we aren't using yeast to make it, we're actually using live tissue. And we actually aren't using human tissue in this for the simple reason that humans don't make enough of this receptor by mass for it to be, I guess, feasible to really use human tissue, meaning in order to collect enough receptor from a human sample, we need to use a lot of tissue, and it just wouldn't be feasible. It isn't feasible to do in that way. So as a lot of other people do, instead of using human tissue, we're using the tissue of Tetranurus californica is a scientific name, or the Pacific electric ray, which, what a cool dude, am I right? But these guys, they have electroplaque tissue where they have a lot, and I mean a lot, of nicotinic acetylcholine receptors, and they're used to make a current, or basically shock I don't know, whoever electric rays don't really with on that day, I guess. But they got a lot of them because they use it to make zaps, make people go away from them. So we just basically take that electroplaque tissue, and that's where we get our acetylcholine receptors from. And so just looking at the differences between a ray and a human acetylcholine receptor, I'm just going to look into some of the nuance here. But essentially, humans and rays have the same subunit composition, except for the fact that humans use an epsilon subunit instead of a gamma subunit. So looking back here, we can see at this green subunit here, in humans we have epsilon, whereas rays will have the gamma subunit. Humans actually use the gamma subunit up until 2 weeks post-birth. Up until that point, for the rest of your life, you're using an epsilon subunit. I'll mention that kind of later on as well. But even the fact that humans and rays share 4 of the 5 subunits, even this one that is different, you're going from gamma to epsilon in humans, isn't that big of a deal. And I'll kind of talk about it later, but the ray gamma subunit is 50% similar to the human epsilon subunit. And by similar, I'm not going to mince words here because this means a very specific thing. This means that at 50% of the residues in that subunit, at 50%, so in half of these spots, the amino acids line up perfectly. So if there's a valine at the 200th amino acid position in a ray, it's also going to be valine in humans. That means it's similar at that 200th spot. And since we have 50% similarity, that means 50% of these amino acid spots are completely identical. But looking at amino acids again, just looking at the 20 canonical amino acids, what you can tell is that it kind of depends on, even if you have a 50% similarity, if you have isoleucine, which you can look at the very bottom here, at the hydrophobic amino acids, if you go from isoleucine to leucine, there really isn't that big of a change. You know, they're chemically very similar, so as in the way that if you have hypothetically, if you have in the gamma subunit for the ray, if you have an isoleucine, and in the human epsilon you have a leucine, you know, they're not similar in that one point, but it's also like, it's similar enough to where they're probably going to have the same function. So just because it's 50% similar, that doesn't necessarily mean that only half of it is the same as the human receptor. Also because receptors are really important, so you don't really want to mess with their function that much. So you can see that these structures are very generally well conserved, even though rays and humans haven't had a common ancestor in quite a while. But tangent aside, we're using electric ray electroplaque tissue to get these receptors from. So basically what you do, you just take these cells, you lyse them, you cut the membranes up, and then you centrifuge them, and this will separate out all of the different parts of the cells into different layers. You take the layer that has the receptors in it, and then there's many different purification techniques, but the one that my lab used is size exclusion chromatography. So similar to HPLC, you're using a column, but it's filled with something very different compared to the hydrocarbon tails. We're looking here, this image, we're looking at the size exclusion chromatography column. You can see it's filled with these porous beads, which they're just of like a very random size, what size these pores are in the beads, but you can buy columns with different size limits. And in this case, basically the whole point here is that if you have something that's smaller, it's going to be able to fit inside the cracks of these beads, and it's going to kind of get stuck in there, just like aimlessly wandering around. The green John is like a little bit bigger, so it can't fit into everything that the red can, whereas blue is kind of massive. It doesn't really fit in anywhere. So when you're going through all of these porous beads, naturally the biggest I guess part of your sample is going to come out first because it doesn't fit into nearly any of these beads, you know, there's nothing to explore, so it just kind of comes out first, whereas all of the smaller stuff is going to by necessity of its size is just going to get stuck in all of these parts where all of the bigger stuff cannot fit into. So just like looking at a short GIF of this, you can see how if you looking at these like blue johns, they come out a lot earlier than the green johns, and the red ones are all the way at the very end. But that is not it. We have the purified receptor, but there's still one more thing we need to do before we can start doing cryo-EM stuff, and that is we need to put a nanodisc around the transmembrane domain of the receptor. And what a nanodisc really is just you have this MSP protein, which is kind of holding together this small collection of lipids, which a lipid is basically just something with a polar charged head, and then its tail is going to be a hydrocarbon or something that's non-polar, doesn't like interacting with water. And the reason we're adding this is that these receptors are normally embedded within the membrane bilayer, and if we didn't add these nanodiscs or lipids around them, then we'd be exposing certain sections of the receptor which are used to being, I guess, surrounded by lipids. They're used to not interacting with water. So if we started doing our samples without these nanodiscs, then even if we had this toxin binding and we could see how this stuff is interacting with each other, it isn't physiologically relevant because we know that receptors have to be within a membrane. That is how their transmembrane domain is the happiest. And one way to really look at this is through the hydrophobicity of these individual residues, which I'll show you a video here. I'm just finding it. Yeah, just like spinning it around, we can see that there's this big stretch in the middle section. That is where all these hydrophobic residues are. And these are all residues which aren't polar. They don't have any electronegative atoms, so they don't really have like any polarity to them. They're kind of just happy chilling out, and they're very happy to be chilling with the hydrocarbon tails, which also aren't charged or anything or have any polarity. They like chilling with the hydrocarbon tails of the lipids. So having those lipids there kind of makes that middle section of the receptor happy. So it's in a state where it would naturally be in, where it naturally functions, which is in the lipid bilayer of your cells. So that's why we have to add this nanodisc there. And then you can also see, forgot to mention, but on the extracellular portion, as well as the kind of skinnier chunk at the bottom, the intracellular portion, which is where they're just like the portions, they're blue since they like interacting with water. These are going to be charged residues. They're going to have an actual charge. They're going to have some sort of polarity where they have, I don't know, maybe some like oxygen or nitrogen, which can do some sort of hydrogen bonding with the water, so they'd enjoy being in that type of environment. But now that we have the nanodisc on there, it's finally time to delve into what is cryo-EM. Alright, so it's finally time to finally delve into what is cryogenic electron microscopy. So called cryo-EM for short. Cryogenic just basically means that we're freezing stuff before we're using an electron microscope to look at it. So before we really talk about how this works, I'm just going to mention just a little anecdote about why we even have to use electron microscopes and why light microscopes aren't useful in the case of looking at how this toxin interacts with this receptor. So the reason for that is that if you're thinking about light, at least visible light that is, that we're able to see, Visible light ranges from roughly 400 nanometers to 700 nanometers. That's the wavelength of light that we're able to see, with 400 nanometers being the shortest wavelength, similar to like a purplish violet color that we'd be able to see, right? But as I mentioned kind of earlier, the size of this receptor is only 16 nanometers by 8 nanometers across the top. So because of that, even the smallest wavelength of light that we could possibly see, which is like a violet color, is 400 nanometers, and that is substantially larger than the 16 nanometers length of the receptor itself. Now, using the Nyquist limit, it basically tells us, I'm just summarizing here, but in essence, the Nyquist limit tells us that the resolution limit of what wavelength of light you're using is roughly half of the wavelength, as well as includes what type of aperture you're using. But if you're using 400 nanometer light, then your maximal resolution of stuff you can make out is something that's 200 nanometers or bigger in size. And so because of that, that is the reason why we can't use a light microscope to look at this stuff, because simply the light is too big for the thing that we want to observe, where essentially what's happening is we're looking to see how the light first, we know what it looks like before it hits the receptor, but then once we're tracking on the detector in the back, what's actually happening to that light. Since the light is so much bigger than the receptor itself, you don't really get any meaningful change in the phase of the light, so you can't really tell what that light really ran into. A pretty decent example that I could give is if you think about human hair, which instead of, what is it here? Human hair is roughly 20 to 180 micrometers, so it's many orders of magnitude bigger than what a receptor width is. It's like thousands of times wider. A human hair is roughly, let's just go with 20 micrometers in diameter. But let's say you want to image that hair with a radio wave, which radio waves can be like a meter up to like some kilometers in distance for that one wavelength. So if you think about, you're holding up a piece of hair and you have a radar going by it, since that piece of hair is so thin and so small, there won't be any interference with the way that light propagates. There's nothing really, that hair is so small and meaningless, it doesn't stop the wave from continuing doing what it did before. So if you had some detector, this side here, you have radio waves going through a piece of hair in the middle, there wouldn't be a meaningful difference between the radio wave before and after. And that's the same thing that happens with light when you're looking at really small structures like receptors. And another maybe more intuitive way to think about this, instead of thinking of light as a wave, we could maybe think about it as a particle. So pretend that you're out with a friend and you really want to make a self-portrait. So as any reasonable person would do, you go out in front of a board and your friend takes a paintball gun and just shoots a bunch of paintballs and just goes in a line and completely covers everything in front of the board, including you. But then when you step away from the board, you're gonna have these particles that hit all around you and trace you out perfectly. But that only works because these paintballs are a lot smaller than the thing they're actually shooting over. So if we think about this large wavelength of light, for example, a 400 nanometer violet wavelength, and you're trying to take a picture of this receptor, since the light is so much bigger than the receptor, it's in essence like if you were to step in front of a board and then your friend takes out like a unbelievably massive paintball gun and the ball that he's shooting at you is like 70 feet like in diameter, you know? Like that paintball is just gonna completely obliterate you, and on the end you won't even see anything. There's just so much overwhelming force that it doesn't matter that you're standing in the way of that 70-foot paintball. It's going to cover the entire back of the wall no matter what you want to do. So there really isn't enough nuance to be able to understand how that paintball's trajectory changed because something was standing in front of it. So because of that, and also just to mention, that's kind of a misleading, like the paintball analogy is sort of misleading just because we're thinking about light as a particle, where in this case, when we're doing electron microscopy, it's really more important to think about the electrons as waves, but it isn't too, I guess, pertinent that we get hung up on that for sake of just being able to explain this complex topic to somebody who doesn't have that much experience with like chemistry or physics of that sort. So now looking at like I guess the range of wavelengths here, we're looking at the spectrum of light, and what we can see, I put on two labels there, two images. We can see that the visible range of light, as I said, is from 400 nanometers, which is like a violet color, all the way over to 700 nanometers, which is a reddish color. And you can see I put the receptor there right around roughly what type of size it is, which is 16 nanometers tall and 8 nanometers wide. So it's right around the wavelength that X-rays would have. And then when we look at electrons wavelengths are completely dependent upon their momentum for their wavelength, and you can know that based on the Broglie, I don't know how to pronounce his name correctly, but basically the wavelength of an electron is equal to Planck's constant over its momentum. Mass of an electron stays the same, so if you change its velocity, you can change its wavelength. So looking back at this diagram here, the electron microscope that I use to obtain my images accelerates these electrons using 300 kilovolts, and it gets them moving at such a speed where they roughly have a wavelength of 0.002 nanometers. So this is many orders of magnitude smaller than the size of the receptor, and that's a good thing, because if we're thinking about the paintball analogy again, we're going from this really freaking massive paintball, like the size of a house that's being shot at you, which is kind of like if you were the receptor standing in front of this board, this 30-foot paintball is like if we're shooting light at you. It's really not useful. You don't get a good imprint on the back of the wall. You don't really see what's there because it's too small. But then when we start using electrons, it's like, imagine now instead of using like something that's the size of your house, you're now using like BB pellets, which obviously I don't recommend shooting BB pellets at your friend, but if we, for I guess sake of the analogy, let's say you were impenetrable to these BB pellets, you could shoot a straight line across the person standing in front of the wall. And because these BB pellets are so small, they give really good precision as to how your body looks, and it can really outline you very well. And that's really the point of using these electrons. They're really small, and they interact with the receptor a lot more than light does, so they can give us a lot better resolution. But now thinking about how electrons are even being shifted when they're being fired at a receptor, what you can think about, or what is thought about, and how it's actually detected that these electrons didn't just go straight through ice and actually interacted with the receptor or something like that, is you can look at the phase of that electron to see if it's different from it before it interacted with the sample and after. So essentially, I'm showing this GIF on here, and what you can see is we have these two waves that are right on top of each other, and then the one in the middle is the summation of both of them. And so you can imagine that this red wave at the top is basically like the electron before it actually interacts with the sample. This is right after it's fired out of the microscope. And then the green one on the bottom is the one that you detect after it's already gone through the sample. And so what you can see is if you have these electrons look the exact same, meaning they're in the exact same position, you'll have constructive interference in the sense that the wave in the middle is going to have a really big bulge, because both of those two waves agree that they're the exact same. So that means you just add everything together and it becomes a bigger version of itself, I guess. But what happens if you interact with something, and since the wavelength of this light is a lot shorter than the thing that it's interacting with, it's possible that this electron gets wound up in the receptor somehow, and when it comes out on the other side, the wavelength or the phase of the wave that it started out as, because the wavelength I think stays the same, but the phase can change based on the way that it interacts with the receptor, and that's what we can track on the bottom of the detector to determine whether or not this electron went just completely straight through in a straight line, or if it took some other path to the detector. And thinking about this kind of more in a classical context as well, because it is kind of tough to think about what it even means to have a phase shift in a wave. Just a classical example, imagine that we're thinking about this electron as a Voyager 2. So I'm showing this GIF on screen here. And what it's showing is this is Voyager 2 as it, I guess, is leaving our solar system. So what I want you to think about is imagine, let's just for simplicity's sake argue that once Voyager 2 left Earth, it didn't accelerate at all. It stayed at the exact same velocity. It did not increase its speed at all, and just kind of kept going as it is. And then, as you can see, when it goes around some of these planets, it starts getting flung around and it gets shot off in a different direction. And so the way we can think about this in terms of how this relates to electron microscopy, think about Voyager 2 as the electron after it gets shot out of the microscope before it interacts with the sample, right? It's on some very specific path, and if it just kept going in that path forever, you'd know that there was nothing in between Voyager 2 and its final end goal, right? But looking back at the GIF, what we can see is if you were to track Voyager's location from beginning to end and you didn't know where any of the planets were in relation to Voyager, you could kind of retrofit based on the path that it took. You would know there had to be something of mass around there to influence Voyager 2 to move in that specific way. And this is very similar to what's happening in electron microscopy, where you're shooting an electron at this massive structure of thousands of amino acids. All of these amino acids are made up of a bunch of protons, right? And electrons, they're negative, protons are positive. So through Coulombic interactions, these electrons will kind of like Voyager go around, and if they feel influenced by one of these protons, they're going to get curved on a different trajectory, which can alter the phase of the wave, or in this case, since we're thinking about a particle, it will basically just slingshot this electron into a different path than it was going to take if it just went straight through the ice. And this path that it takes through the receptor could be very chaotic, but if we see the electron at the end of the detector, then we can determine based on what it looked like at the beginning what type of path it took and what had to be in its way in order for it to get to that point. That's just kind of like the essence of what's happening in an electron microscope. I hopefully did that justice, but enough time, enough waiting. Finally, I'll show you the microscope itself. And holy mama, that is one big boy, you know? I'm a pretty tall dude, and this thing is like, it's pretty massive. Like standing next to it, it's like, this is like 10 foot, maybe 11 foot tall, just John, and it's just kind of standing there. It's really intimidating. It's like, has its own whole room to its own. I don't know. I just think it's really cool. But how this thing works, as I mentioned, you're using 300 kilovolts of electricity to accelerate these electrons. So this is a process kind of similar to mass spectroscopy in the sense where you're taking an electron gun and you're just generating a bunch of electrons, and then you're using an even more powerful electric field than in a mass spec, using 300 kilovolts to accelerate these electrons to close to the speed of light. And then once you've accelerated them this fast, then you use magnets to, I guess, condense those electrons into a really tight beam, because you want a really thin, skinny beam, because the holes on the grids aren't really that big, so you want all these electrons condensed into a really small space. And that is essentially what is happening in this electron microscope. You take electrons that are generated, accelerate them to almost the speed of light, and then you bend them using a magnetic field so they're all going in one straight line down through the sample. And looking at kind of just like an internal of what's going on in the microscope here, this is an image from Penn State, I think. I'm pretty sure it's the same model as the one that I used, but what I just want to point out here is like, goddamn, that's like pretty gnarly, right? That's a lot of complex like wiring and stuff. Like, hats off to the engineers who were able to make that happen, because I sure as hell do not know what most of those wires do, and it's just kind of impressive to look at. But now looking into the lens setup, I don't really have anything of substance to add here other than it's pretty complex. Looking here, we can see that there's 3 condensers, there's other types of stages, and then out at the very bottom, you can see, where is it? Like in between the red arrow at the very bottom and the other John, that's where you're gonna be getting like the image being taken. And yeah, basically that is the essence of electron microscopy. I'm gonna briefly just run you through the process of what I actually did before we actually get into the data processing. So you're basically taking this copper grid, and on top of it, you're taking just a little bit of water with, I put in the purified receptor with the nanodisc, as well as the toxin itself. So those are in the water. You put that on the grid, and you try and get as much of that water away as you can. Then you freeze it in liquid ethane, cooled by liquid nitrogen, at negative 180 degrees Celsius. And from there, you can use the electron microscope to get data. Then you do image processing, and then finally model building, where you're taking the electron density map that you found using the microscope, and then since the receptor of the nicotinic acetylcholine receptor has already been solved previously, so we already know what it looks like. But since it's interacting with a new toxin that hasn't been looked at yet, is going to give a novel new like form as to how this receptor shift happens. So we can just use a plain Jane nicotinic acetylcholine receptor channel and fit it inside of the EM density map to see how this receptor changes when it's bound to by this toxin. But looking more in depth into the grids here, just to give a better understanding of what we're actually holding our samples on, it's a copper mesh kind of, with many grid squares, and all these grid squares are filled with a bunch of holes, and these holes are where our samples are going to sit. So looking here at this image, you can see there's the grid here. It's filled with many of these grid squares, and each of these grid squares is going to contain a bunch of tiny circular cutouts, where we hope that the water with our sample is suspended such that there's nothing in between the top and bottom layer of that ice sheet. And so the reason we freeze at negative 180 for like one of the biggest reasons is that it just reduces motion. And something that you'll notice is that even at negative 180, there's still a lot of motion that happens at the molecular level, but since we're freezing it at these really low temperatures and keeping the samples at negative 180, that means that there's a lot less motion, there's a lot less noise, so we can get a cleaner idea of how stuff is actually interacting. And now, when I actually made this sample, you have the grid. I froze the toxin and the receptor in the grid squares. Now it's time to actually look at it using the electron microscope. So that's an image I'm showing here. This is looking at the entire grid, and you can kind of see the whole circular like shape of it, and that's the entire grid. So this is not that high of magnification at all. But what you can see is that there's a lot of variability in these grid squares, where some of them have like black spots just kind of hanging out. This is crystalline ice, and that is why we try and freeze at -180, is to, instead of having ice crystals forming, you want just a uniform sheet of water that all looks the same, so that if an electron goes through, there's no interaction at all. Because if you have ice crystals in your sample, then those are going to shoot the electrons off into many different directions and kind of lead to not being able to use that data, just because the electrons aren't cleanly being shot through the ice. So there's some sort of nuance there to when we're making the samples. We need to make sure that there's just a very thin layer of ice only covering like those holes through the grid. And we just want there to be like a thick enough layer so that the actual receptors aren't popping out of the water, but thin enough so that you can actually have electrons fully shoot through. Because what you can see in this image here is that there's kind of like this dark spot, like this black spot, where you know that the grid is over there somewhere. But what happened is that there's just a layer of ice that's too thick over this region so that electrons can't cleanly get through to the other end. So half of the grid is basically unusable. There's still lots of grid squares that we can use to collect data. Now, looking at the actual grid holes themselves, this is just an image. You also see the, I guess, steering rig that you use for the microscope down there at the bottom. There's hundreds of these like circular cutouts in one grid square, and there's many grid squares on one grid. And basically what we're doing here is we're just labeling specific grid holes for collecting data afterward. And then here we are looking at the actual, like what a raw image from an electron microscope would look like. This is not an image that I took, but it just gives you the sense of, you know, kind of as you'd expect, doesn't look as, I guess, sensational. There's only black and white, you know, but you can actually spot out like individual proteins at this level, which is really sick. Like, as you can see in the image here, we have like this, I don't know what this protein or structure thing is, but you can see with the blue circled, like, I guess, thing in the raw image there, that's supposed to be this structure on the right. And something else that I'll bring up here that'll be kind of pertinent in the next section is that when you are taking these images, there's kind of a fine line you have to play between how much defocus you want. And by defocus, I mean that when you have a lens, you know, it'll bend light and it'll focus on to some specific focal point. But the thing about looking directly or having your sample directly inside that focal point is that you have really no contrast at all. So you can't really discern like white from blacks, and it all kind of mixes together so you aren't actually able to see anything in the sample. And it's not really good for us when we're just trying to spot stuff that's in the sample, but it's also really good because at that focal point you have the cleanest amount of electrons going through, and you have the cleanest, I guess, electron scattering going on. But if you want to pick out particles that you think are interesting and you want to look at further, you can't even pick out any particles because there's no contrast in the image. So as we're seeing in this image here, there's some amount of defocus applied, meaning that when you're actually taking the sample, let's say the focal point is like down here, you might have the sample like, I don't know if it's slightly above or below, but you're slightly not, like you're not at the focal point to give you a little more contrast so you can actually see some of the structures that are in the sample there. And that's just kind of something you got to toy with in order to get good data, because the closer you are to the focal point, the better the image you get, but the farther away you get, the easier it is to actually spot the stuff that you want to look at. So Yeah, that was all of just the intro to cryo-EM, and now we can actually get into data processing. All right, so we're finally at the data processing. We are very near the end, and recording this, I apologize for how long this video is. I didn't realize I had that much to say, but let's just jump in, not make it any longer. First thing, we're just gonna look at this image here, just like a basic, I guess, idea of what I've already said in the previous part, but just to reiterate, what you have is this very thin sheet of ice where your sample is located. In this case, I have my receptor with the nanodisk as well as the toxin floating in here. You're shooting electrons through it, and at the very bottom you got a detector which can detect these electron strikes. So if you have an electron just going through straight ice, it won't feel any, or it won't interact at all, and it'll just go straight through. However, if you have something in the way, such as a receptor, you can have electron scattering, which will lead to that electron not landing in the place it would have if it just went straight through the ice. And then you can reconstruct that scattering of electrons into a 2D image of what the thing that scattered the electrons must have looked like. And so that's kind of what's being shown at the bottom here, is you're collecting a bunch of 2D images of the thing that these electrons are scattering off of. And the goal is to get as many 2D pictures as you can from all angles possible, because then you can use these 2D images to then reconstruct a 3D image. So for an example here, let's say for whatever reason, I wanted you to reconstruct this speaker. I can give you, like, for example, here's one image, here's another, and then there's another one right there. And from that, you have 4 images that are 2D of the speaker. But based on those images, you should be able to reason that this speaker is a pill shape, right? But then, crucially, since I left out the back, if I told you to make the structure of the speaker from just those couple of images I gave you and left out this back section, you wouldn't know that it charges on the back here. Very similar to in cryo-EM, it's really crucial that we get 2D images from all around of this receptor bound to this toxin, because this isn't a symmetrical molecule. There's 5 different subunits and they all look different. I mean, 2 of them are the same, but there isn't that much symmetry there. So we need to see this thing from all angles so that we can reconstruct a better 3D image, because the more 2D images we have, the more sure we can be that the 3D image we come up with at the end is the correct thing. And also important to note is there's kind of a resolution limit as to what I want to do, meaning that if you just want to get the general shape of something, then you only need like a 10 to 30 angstrom resolution, which isn't really all that great. It gives you the general shape of what's going on. Then if you go in between like 5 to 10 angstrom resolution, that's when you can start seeing alpha helices and beta sheets and these type of secondary structures I mentioned at the very top of the video. And then at the very end here, sub 4 angstrom resolution is what you need to really be able to see individual side chains and see the individual amino acids as they are in this structure. And that's really important because what I'm trying to look at is these individual amino acids on the toxin and the receptor that interact with each other to mediate this antagonist interaction. And so just looking at kind of a flow chart of, now that I've collected all of my data, which is roughly 4,600 movies, or basically just 4,600 recordings of some sample using the electron microscope. The first thing, here it says we have to do particle extraction first, but really the first thing you got to do is motion correction. And this is all stuff that's done by a program for me. I don't do any of the calculations myself. But you have to do this type of motion correction, not because receptors and toxin, even at negative 180, there's motion, but they aren't really moving around that much. The type of motion we're correcting for is that, as I mentioned, we're shooting electrons near the speed of light at this sample. So since these electrons have a lot of energy, they cause this ice to expand. And as you remember, we had like this really thin disk of ice, but it actually starts expanding and kind of swelling when you have electrons shoot through it. So you have to correct for, let's say you have something like a receptor in the very middle here. You have to account for how much it moved when that ice expands. And that's one of the first steps you do with the data. The next step, as I mentioned before talking about the defocus and contrast, you have to estimate the contrast transfer function, which is basically a way to model, it's basically a way of just telling you how much signal is in your data and what type of resolution you can really see out to. I honestly don't really understand the CTF estimation too much, but that's something else you do before you actually dissect the data. But once you do all that sort of correction stuff, you have a good amount of data to work with. What you do is you take all of these images which have a decent amount of defocus so that you can actually see in the raw images like these shapes that are appearing in there. The first thing I do is I tell this program, hey, I'm looking for this receptor, and specifically this receptor is 16 nanometers in length and 8 nanometers across. So I just say, look for anything that it fits that size and show me what you I guess can see. From that, I think I was left with like 8 million particles or something like that. From there, we can tell the computer to do a 2D classification, which you can see is one of these steps on here. And basically what you're telling the computer is, I want you to sort out all of the particles that you just gave me, the 8 million particles. I want you to sort them into, there's 30, I only want 30 bins. Pick the most frequently seen particle and just put it in a bin. And if you see something like it, put it in the same bin. So you can have bins of all the same particle, but they're all looking at the particle from different orientations. So you're just trying to pick the bins of the particles which look the best, and also pick the ones which have all of the orientations you like to see. You want to have some from the side of the particle as well as a top-down view. So then you can use these bins to then more efficiently look for other particles that are like them. So you can use these 2D classes to look for even better 2D classes. And then once you have enough of these images, you're confident that all of the 2D images you have resemble the thing that you're going after, then you can start using those 2D images to reconstruct a 3D image. So the whole goal through this data process is to really remove junk data. So for example, a way to describe this, I guess, is imagine that like with the speaker I asked you to reconstruct before. Let's say I sent you just this front shot, this side shot, top, this side, bottom, also the back. You have everything you need. But then I also accidentally take not butt dial, but I take like 30 butt photos of just like pure like wiped out, like just unusable, like nothing burger photos, and I send you 30 of those along with the 5 of the speaker that I sent you. This computer isn't smart enough to be like, hey, there's 30 photos that have actually nothing here. It will say, this must be part of the actual structure. So then when it goes back into the actual reconstruction, it'll get confused and you won't actually get in reality what's happening. So you're trying to toss out all these junk images that have nothing in them, and you only want these 2D images that show a very clear receptor binding to some toxin. And yeah, that leaves us with, once you go through that whole process of tossing out all these junk particles, I'm left with roughly 100,000 particles to work with. and then we can start looking into the actual EM density map that I made. So looking here, the EM density map itself has a resolution of 2.82 angstroms, and the colors here are showing the local resolution, so the residue or the resolution at each residue. And what we can, or I guess just to briefly describe what we're looking at here, this EM density map, a way to think about it is all of the stuff that's filled in, because this is like one big huge structure where anything that's like inside of this density map is an area of which the electron cannot reach, because as I talked about in a previous video, electrons can fall into the nucleus. There's some limit as to how close they can get to a proton. In this case, it's kind of like drawing the limits as to where electrons flew by before they started getting drawn into by Coulombic interactions of some like proton in this structure. So basically just seeing a map of where these electrons got scattered. And that's what we're looking at here. But looking at the specific local resolution, what you can see is that most of the structure is really well resolved, roughly at 2.5 angstroms, especially when you're looking at the very right one, which is looking at basically just the receptor cut through down the middle. You can see that a lot of these residues have a really nice resolution around 2.5 angstroms. And then you can, looking at the one on the very left, you can see that some of these structures, like towards the outermost part of the receptor, have closer to like a 4.5 angstrom resolution. And so just something that I'll bring up here, but I'll mention later as well, is that when you're amino acid buried in the core of this receptor, you really don't have a lot of motion going on. The most you might do is like maybe move a tiny bit when the receptor opens its channel, but a bunch of these amino acids are kind of just stuck in place, and they're just there as like a scaffold. And because of that, if these amino acids don't ever really move, that means if an electron were to happen to scatter off of that area, it's going to scatter in the same way every time, leading to a better resolution there. Versus if you have some sort of amino acid near the outside, which maybe moves around a lot, likes interacting with water, every time the electron scatters off that amino acid and it's in a different orientation every time, you're going to have lower resolution because there's not as good scattering at that area. You aren't as certain of the position of that amino acid because it moves more. So this local resolution is kind of telling us what is better resolved, but also telling us what doesn't move as much in the structure, kind of, but not really. But it's a good enough way to think about it for now. But from there, we can tell based on that local resolution I just showed you that structure is decently well resolved, and we can start looking for where this toxin actually binds. So this is what I'm showing you here in this image, is you see this grayish like blob, that's the EM density map. And I've circled in pink. You can see that there's the ribbon structure, or just the carbon backbone of the receptor, which I superimposed and put inside of the EM density map. So as I showed you earlier with like the rotating receptor, like that structure itself, that is what I put inside of the map itself, because in theory, the model of the nicotinic acetylcholine receptor and the EM density map we get should be the same. They should physically look the same, although the orientation of some of these amino acids might be a little bit different. And so that's what we're starting to do right now, is the model building, where we take the receptor, the model of it, and fit it inside of the experimentally determined data and see how the receptor differs from the data to the apo state or the closed conformation. God, I sounded like Robert F. Kennedy Jr. there. That's my bad. But looking more at this picture, what you can see is there's two different density thresholds that we're looking at. So on the left is a really like high density EM threshold. And what that basically means is that on the left, this is where there's a higher density of electron scattering events, whereas on the right, we're looking at a lower density EM density map, meaning that there's less amount of electron scattering events that happen there because, among other things, there's a lot more motion in these areas. So what I'm circling here is you can see in these red circles, these all correspond to sugars that like interact with the receptor, as I mentioned before, but they have their own EM density map associated with them because they can also scatter electrons. So what our goal really was to do was to take this receptor that has all these sugars on it, put it inside of the map, see which of these blobs correspond to sugars and stuff that should be on the receptor, and try to find something that doesn't really fit anything that we know should be in that spot. And that's what we have circled on the green there. That is where we propose that OSC1 is binding. And so now looking at I have to pull this up real quick, so give me 1 second. But yeah, looking at this video here, what we can see is my cursor is looking over that, or going around that point where the toxin is. I'm going from a high density to now I'm turning into a lower density map, looking at the less dense regions. You can see there, I'm kind of circling the toxin, which showed up as the lower density region since it's more mobile. Then you can also see I'm kind of circling around. Down at the bottom there, we have the nanodisk, which you can see those lipids, and also I pointed out a sugar that's up at the top there. And now that's basically just like showing you going from the higher density strikes, which are those that hit regions that don't move as much, versus the lower density EM map, which is showing you regions that move a lot more. So for example, the nanodisk, where these lipids are moving around very fast. Yeah, so that's basically how we got to identifying where this toxin was binding, you know? And now after that, what we can do is we can build a focus mask, which is basically a region that we're specifying for this program to say, hey, this is the region of interest. So you can see this is the receptor like by itself, as well as us placing the toxin where we think it's going to bind. And you can see this blue blob next to it. And this is basically just this region that we made. It's just like basically telling the program we fit that blue blob over top of the top region of the receptor, as well as covering the toxin there. And basically what we're telling this program is to now, instead of doing a 3D classification, or instead of a 2D classification, where we're looking at a bunch of different 2D images that happen the most often, we're now doing a 3D classification with only 2 classes. So in theory, it's showing us, it's splitting up, it's classifying these particles based on the way they look in a 3D, but only in the region that we showed them, which is the blue mask that I made. And so from there, it can lead to a better 3D reconstruction, which I can show you now. We're looking at this, they have said, has a resolution of roughly 2.94 angstroms, so a little bit lower. But what you can see is that the local resolution looks a decent bit better. And it's basically just giving us, circled in black there is the toxin. And what you can see, it isn't really super well resolved. Yeah, it's roughly like 6.5 angstrom, maybe like around 5, or yeah, something like that. Not really super awesome. But something that's also interesting to point out is when we look down the middle there, we saw that there's actually something hanging out in the middle of the pore, which, if you remember at the very beginning of the video when I was talking about AlphaFold, it kind of brings back that potassium channel, where AlphaFold thought that OSC1 could potentially bind the middle of this potassium channel. So it's interesting that we see that OSC1 is binding the middle of this nicotinic acetylcholine receptor, or potentially is. We still don't know for sure. But another thing to point out here is that we only see OSC1 binding to the alpha gamma site and not the alpha delta site, which was predicted by AlphaFold. So it's just kind of something, like, I'm hesitated a little bit to call AlphaFold awesome in the beginning. That's just because, like, it's a great tool, but it doesn't really replace scientists in any way, shape, or form. Because as you can see, with the way it predicted it bound to the alpha delta site. It does not. It predicted it bound to the alpha gamma site, which, sure, yeah, they got that one. But it also didn't predict that OSC1 binds in the middle of the pore. So, well, yes, it is awesome to think about all these AI tools that are flooding out. It isn't necessarily they're going to replace us. It's more of making work easier for us. And scientists, I don't think will ever, or hopefully not ever, be replaced. That would kind of suck if they do. But now that we have a pretty good map made from that 3D classification, we have a pretty decent map, and now we can actually start trying to, because what the program that I'm showing you, Chimera X, it does a pretty decent job at aligning the model with the map, or basically the ribbon structure of that receptor and the toxin can get aligned pretty decently well with the EM density map in Chimera, but you need to use a more specialized program to really be more finicky with it and actually work out all the kinks. And that's what we use this program Coot for. And basically, one of the most important things that it looks at and that we fix are what are so-called Ramachandran outliers. And so Ramachandran outliers are basically, you can have this Ramachandran plot, which is looking at two very specific bond angles, and I'll show on screen here. What we're looking at here is this amino acid. So we have this amino group on the left, the carboxyl group on the right, and then we have the alpha carbon there in the center attached to a hydrogen, as well as the R group. And so the orientation of the alpha carbon with respect to the amine group can be notated as phi, and it can go from -180 to +180. And the same thing with psi, but instead of the alpha carbon and amino group, it's the alpha carbon and the carboxyl group. So it's basically just a way of writing down what the angles are for every alpha carbon of every amino acid in the structure. And what we can see, looking at this Ramachandran plot, these secondary structures have a very specific bond angle that is needed for them to work. And that is, these secondary structures require these amino acids to be positioned, and they require those bond angles to be positioned in such a certain way so that those secondary structures can be stable. And in the case of this nicotinic acetylcholine receptor, whose structure has already been solved, we know which amino acids should be in which secondary structures. So when we input this into the program Coot, what I can show you here is the blue is signifying the EM density map, and all the yellow, that's basically all the amino acids in the structure, and we're trying to fit them into the map as best we can. And you can also see the Ramachandran plot on the right there, and you can see we got some triangles as well as red circles, which signify outliers, where these are amino acids we know should fit in these secondary structures, but have bond angles which would not be conducive of letting them be in these secondary structures, if that makes sense. We need to go into the actual structure itself. This might be a better image to show. You can actually see these amino acids fitting into the EM density map. So if we ever come across these Ramachandran outliers, what we can do is we can basically force these amino acids to take up a different conformation or spin, rotate their bonds a little bit so that they fit into the EM density map a little bit more nicely. And that's something we can do in Chimera, so that's what we use this program Coot for it. But then, other than these Ramachandran outliers, there's also these things called rotamers, which are very similar in nature. They're basically, like, looking here, you can see the angles phi and psi. Those are going to be what are found on the Ramachandran plot. And I think that's chi there, we're looking at, I honestly couldn't, I don't know what amino acid that is. That's really embarrassing. I'm sorry, guys. But you have some R group, and you have these three chi angles, chi 1, chi 2, chi 3. And basically, it's just showing that for all Ramachandran outliers are just looking at the backbone, or basically the ribbon structure of the receptor as well as the toxin. And rotamers are looking at only the R group and seeing if all of the bond angles of the R group make sense. So basically, we're just going through the entire structure of this receptor, and we're double checking that the bond angles that are seen to fit into the EM density map the best also were conducive to what we know bond angles should be possible based on our current understanding of the laws of physics. That is a very short, roundabout way of saying what we're actually doing in this program, and we're trying to fit this model even more neatly into the density map. And so here's another image just showing all of the rotamers that we had to fix. I think this is even before this was started a little bit. I already fixed some of these rotamers, so there's a lot more that were messed up. You can also, you can see that I fixed some of the Ramachandran outliers here. Then moving on, we can see this is the finalized structure, no more rotamer outliers, and all the Ramachandran plot looks pretty decent. So this means that all of the amino acids in this receptor as well as the toxin have bond angles that fit with our current understanding of how they should be allowed to bend given our current understanding. And that basically leads us to, we now have a pretty decent structure and we can start doing data analysis. With something, before we really hop into the results here, that I should mention is that the results I'm showing you are kind of due to a time constraint where I had to show some work because I was going to graduate at some point. So I was like, okay, I need to have at least some data, which I won't be the first one to tell you this data isn't perfect. It's definitely a rough draft, and I'm currently still working on this project. So don't think that this is like the final say on what happens between OSC1 and the receptor. But before we actually delve into this interaction, just something that I should bring up and is kind of indicative of the fact that this is a rough draft. If we're looking at the connectivity of the disulfide bonds of OSC1 in this finalized structure that we got, we can see that I've circled here the disulfide bonds and as well as color coding which ones should be forming. And as you can see, with cysteine 8, 28, and 14 and 23, all of them are kind of nearby each other, and we know that cysteine 8 should bind cysteine 28, and it kind of seems like that is what's happening, but due to their close proximity to the other two cysteines, it's kind of confusing the program to where when I was visualizing this in Chimera, it kind of thought that all four of them were bonding with each other. This doesn't necessarily negate the findings and is more kind of showing us that we need to further refine our structure in COOT, and just showing us that we probably just need better rotamers, because it's possible to pick rotamers that are allowable bond angles but also don't fit into the density map as nicely. So it is possible that I just didn't do as great of a job of fitting the toxin into the EM density map here. Another way to know that this doesn't negate the findings is that it's previously been known that OSC1 binds nicotinic acetylcholine receptors, and we saw that it bound to at least one site. So we know that the activity of it is conserved, and I'd be surprised if the reason that this came about was because I actually synthesized OSC1 incorrectly. I don't think that's the case here. So with that being said, we can actually move on to actually the main interactions that are holding OSC1 onto the receptor. This is the actual, finally we're getting to the point of understanding how this interaction works. So looking at this here, we can see that there's three different hydrogen bonds, which, just to jog your memory a little bit, hydrogen bonds happen when you have a more electronegative atom, which is pulling on the electrons of something that's less electronegative than it. So for example, oxygen, nitrogen, and sulfur bound to carbon, they all have a higher electronegativity, so they're going to pull those electrons closer to themselves and have a partial negative charge, meaning that when you have water going around, for example, since oxygen is more electronegative than hydrogen, the oxygen is going to be more with a partial negative charge, and the hydrogens will have a partial positive. So you can have these partial charges interact and stabilize, the structure, which these H bonds stabilize the structure of alpha helices and beta sheets, which are secondary structures, but you can also have them stabilize tertiary structures as well as quaternary structures, which we are seeing here. So yeah, you can see there's three different hydrogen bonds being formed here, and the pictures are labeled so you can hopefully spot those out by yourself, as well as a salt bridge, which salt bridge between aspartic acid and lysine. Lysine has a net positive charge and aspartic acid has a net negative charge. So that's probably going to be the most strong interaction of, I guess, in here that's keeping this thing together. Then there's also a bunch of hydrophobic interactions, which, not that they aren't important, but compared to the other three, they aren't as crucial, I'd say. So I'm just going to kind of gloss over them here. And comparing my results to a previously known toxin, alpha bungarotoxin. This alpha bungarotoxin is also an alpha neurotoxin, as is OSC1. And these alpha neurotoxins, all it basically means is they target the alpha subunits of nicotinic acetylcholine receptors and prevent the flow of sodium through, causing paralysis, as we know. So now looking at how they're the same and different, the only thing that is really shared by alpha bungarotoxin and OSC1 is that both of them use cysteine 192, which OSC1 uses to form a hydrogen bond with, asparagine. Yes, that is asparagine 30. Cysteine 192 is bonding to hydrogen bonding with asparagine 30. So I had a brain fart there. And then cysteine 193 also has this hydrophobic interaction that happens. And these are the only residues off of the alpha chain of the receptor that OSC1 actually uses for binding. And other than that, it only really binds to gamma, which, put in parentheses, which chain these amino acids that interact with OSC1 are on. You can see that almost all of them are gammas except for cysteine 192 and 193, which is pretty interesting. Maybe even more interesting than that, something that should be noted, is that this alpha bungarotoxin uses tyrosine 198 to bind, which that's circled in red here. And I guess in green I'm circling the cysteine 192, they both bind both alpha neurotoxins. But in red here, we're looking at this tyrosine 198, which forms a pi cation interaction with positively charged residues. So in this case, we're looking at an arginine here of the alpha bungarotoxin, which is shown in orange. I guess I should have explained that better. The alpha bungarotoxin is shown in orange. The alpha subunit is shown in this weird greenish color, and the gamma is shown in a blue for the alpha bungarotoxin. And what you can see, as I was saying about this tyrosine 198, it forms a pi cation interaction where this tyrosine's benzene ring, these electrons can delocalize, so where they kind of form like negative clouds on either side of it. So you have this delocalized cloud of electron, like let's say you have this benzene ring here, you have this delocalized cloud of electrons on either side. And then if you have a positive charge, for example with the arginine, it can hop in there, and the net positive charge on the arginine can interact with what is kind of like a pseudo charge of the delocalized electrons from that benzene, and those can interact to stabilize binding further. But what is more interesting about this is that tyrosine 198 is one of the key residues that is known to bind acetylcholine. So as you remember, acetylcholine, and that's what I'm showing at the top there, you can see the net positive charge on the nitrogen there of acetylcholine, which would take the place of arginine 36 of alpha bungarotoxin, and that is where it would normally be nested in between the alpha and a gamma subunit. That's one of the binding sites. The other one is the alpha delta, which, as you remember, OSC1 doesn't bind to, which is pretty interesting. But then now going more into kind of other implications of this data, as I mentioned, the receptor we're using is not actually human receptor. It's from the Pacific electric ray, I think, if I'm remembering correctly. But this ray is using the gamma subunit, whereas humans use the human epsilon subunit. So as you remember, these are 50% similar, meaning that at 50% of all amino acid locations, 50% of them are the exact same between the gamma of the ray and the epsilon of us humans. So looking at the other 50% that aren't similar, I wanted to see how many of those that are changed still conserve the function of the one used previously. So looking at those, what we can see is that on the ray gamma subunit, you have aspartic acid 113, which changes into a glycine, which I think it was doing a hydrogen bond before, which now no longer possible. Glycine has no oxygens, nitrogens, or sulfurs or anything, so it can't hydrogen bond. So we lose one of those interactions. But the other two that we can see, we go from I think it's glutamine to aspartic acid to glutamate, and those will conserve those. I'm just gonna go back here and double check. Okay, the very last one, the aspartic acid changing to glutamic acid, those are gonna keep the change of, you're still gonna have a net negative charge, you can still form the salt bridge. This other one can still form a hydrogen bond. So if you still have 3 out of 4 of those major forces that are causing you to bind, there's still very high probability, or there's still, I wouldn't say a high probability, it wouldn't be surprising to see that if we were to use human nicotinic acetylcholine receptors, we'd probably see the same type of binding between the alpha epsilon subunit that we're seeing on the alpha gamma subunit of the rays. And that's really what I'm trying to show here. This image on the right is also showing you the differences between the gamma subunit and the epsilon subunit of humans, and just showing the different amino acids that are swapped out. And yeah, because of the reason that this receptor binds very tightly to just the gamma subunit, it made me think that, because there's still kind of an unsolved mystery of humans are born with the gamma subunit, but then 2 weeks post-birth, we switch to using the epsilon subunit, which binds acetylcholine like 30 to 40 times less tightly and is open for 3 times shorter than this gamma subunit would allow for. All the rest of the subunits are kept the same. So a thought is that maybe OSC1 could be a good toxin to test the differences between the human alpha gamma and human alpha epsilon subunits, to see how their roles play in development, since as we know currently, nicotinic acetylcholine receptors play a role in patterning and innervation and kind of creating your body scheme, for lack of better words, in development. But we don't know why there still is this change in affinity by swapping out these subunits. And just kind of like last little note, as I already kind of mentioned, since this toxin also seems to bind inside the pore of this receptor, it can give us insight maybe into the evolution of this toxin and whether it first had OSC1 bind inside potassium channels, or if it first bound inside nicotinic acetylcholine receptors, but that's kind of like a bigger question yonder beyond the horizon, which isn't super relevant with the amount of data that I have so far. But that is kind of the end of where I ended my honors thesis defense. Since then, I have graduated, been on vacation, and not done a super lot, but I have done some work on this project, and just gonna share a little bit of what I've been doing, I guess. So looking first at something that we found with that second local resolution that I showed you, it was good in that it showed us where this toxin was binding, but we didn't maybe get the best resolution at that site. So what I did, like to move on further and try and improve the data quality, is to make two new focus masks. One where it's purely just hovering over the gamma site, just the outside of the gamma site, as well as the receptor, the toxin, just trying to keep it over there, as well as a focus mask just in the very center of the pore. And that's what I'm showing here in this video. You can see that as it's spinning around, this like cyan, like light bluish color, that's the thing that's covering the gamma site as well as the toxin there. And then you have this deeper blue, which is basically just filling the inside of the pore there. And these are basically specifying the regions for a 3D classification where we want to look at very specific differences and get a better reconstruction, but only in that area. And that's kind of what we're telling the program to do, just to focus on those areas to get a better, I guess, understanding of what's going on there. Because we know that the toxin binds the receptor of the alpha gamma site, also binds in the pore. Those are why we have the focus mask there. So from the focus mask, we can look at the actual EM density maps they produce. So that's what we're looking at here. I kind of picked some weird colors just so you can tell which is which. But basically you have this purple color, which that's just the EM density map with no 3D classification at all. That's just a plain Jane 3D reconstruction. Then you can see the red is the map of the focus mask used to look at the alpha gamma binding site. And you can see it's covering most of this toxin now, which actually looks a lot better than previous maps did. And then you can see kind of poking through the middle there is the green, which is mainly giving us a EM density map for inside of the pore. And what we can do now is, since we have three maps which give us a good view at very different things, we can combine all of them together into one map to give us the best of all worlds, where we can have really good resolution or a really good EM density map of the toxin binding to this gamma site, but then we can also get good resolution of inside the pore without having to sacrifice the resolution of stuff on the very bottom of the receptor as well. And so that's what we're looking at here. It's basically just the map overlaid onto the model of the receptor as well as the toxin shown there. And you can't really see it super well. You'll see it better in the next image that I show you, but it covers this receptor pretty darn well. So looking now at like the actual local resolution, I can't access the program I used to create these maps for right now, just with the case I'm in right now. So I don't actually know the resolution of this image, but we can see the local resolution here. And just something to note is that circled here is the toxin bound to the receptor. And if you remember from the second local resolution I showed you, the toxin was really at around like a 6.5 angstrom resolution, whereas here we're seeing more of like a 4.5 to a 5.5 resolution. So we can see that we have improved resolution on this toxin binding, as well as the pore looks a lot noisier than it did with the previous it makes it a lot noisier than it did with the previous, what's it called? The previous focus mask we use. It kind of gives us a better idea as to what's happening in the core. I don't know if this is going to be that great for actually fitting OSC1 into the pore, but yeah, I still need to use Qt, do some more model building. This isn't the end of this project, but this is the end for right now. I'm still working alongside my principal investigator to get this final structure. I might revisit this topic someday. I hope that this work will eventually get published. That's the goal for right now. I think in the next year or two, it should definitely be in some sort of journal. But if you are one of those crazy sexy who made it all the way through this video, I really appreciate it. I'm glad that you were interested and I kept you captivated for this long. If you enjoyed, I'd appreciate if you left a like on the video. And yeah, I expect to see videos every 2 to 3 weeks. I think I'll be able to hold myself to that standard. But this is Kember out, and as always, I hope you have a good one.